Good morning, dear students. Today in this lecture talk, I'll be discussing with you antibody structure and functions of immunoglobulin A. The purpose of making this video is to understand very easily the immunoglobulin structure A, and I have given all the points. If you refer this particular information for your examination, that will be more than enough. Okay. Now let us go through the antibody structure and functions of immunoglobulin A. IgA, that is immunoglobulin A, which is the second most abundant immunoglobulin and account for about 15% of the immunoglobulin pool, which is next to immunoglobulin G. Okay, because highest percentage is this immunoglobulin G. IgA is named so because of presence of alpha heavy type of chain. Okay, so we have named because name this IgA as alpha type, right? A stands for alpha because in the heavy chain there is alpha type of amino acid sequence, so they mentioned that as the alpha type. Okay, hope you understood here. In serum, immunoglobulin A present in the form of monomer of two heavy chains and two light chains. If you take any immunoglobulin, you could see the presence of two heavy chain and two long chains which are connected by interdisulfide bond. IgA occurs in the form of serum IgA and secretory immunoglobulin A. When this serum immunoglobulin A and secretory immunoglobulin joins together, that time we'll say that as the dimeric form of immunoglobulin A. Most immunoglobulin A accounts in the serum as polymerized dimer joined together by a polypeptide J chain. IgA found in tears and in the mucus secretions of respiratory gastrointestinal tract, breast milk, and in the tears than saliva. IgA concentration, that is 10 to 15 percentage in the immunoglobulin pool. IgA is the predominant Ig present in saliva. Okay, it has four polypeptide chains where you could see the presence of two light chains or of kappa or maybe a lambda, two heavy chains that will be alpha type. The molecular weight is 160 kilo Daltons, half-life 6 to 8 days. In IgA, we can see the presence of J chain. Okay, this J chain basically made up of polypeptide. It means they're made up of protein molecules, right? Polypeptide, where you can see the presence of amino acids which are joined together through the peptide bond. So J chain, which is a polypeptide, which is identical to the J chain which is present in the IgM immunoglobulin molecule. Okay. The J chain molecular weight is 15,000 Daltons, polypeptide chain, which is called secretory components. The J chain which connecting the two immunoglobulin node, that is known as the secretory component, which is secreted by the plasma cells. J-chain component facilitates polymerization of both serum IgA and secretory immunoglobulin A. IgA special features associated with secretory mucosal surfaces. It occurs as a dimer formed of two molecules of IgA linked by a secretory component, okay, or secretory piece, and a J chain, that is the joining chain, which are made up of proteins. IgA, when transported from mucosal associated lymphoid tissue to mucosal surfaces, it acquires there a polypeptide termed as secretory protein and called secretory antibody or immunoglobulin. The secretory piece stabilizes the immunoglobulin A dimer against proteolysis and is a single polypeptide chain of molecular weight 70,000 Daltons. Okay, the secretory component which is of 70,000 Daltons, whereas J chain is different. Okay, IgA is synthesized by plasma cells in lamina propria of mucous membrane and dimerized intracellularly by secretory chain and in the presence of J chain. So here you can see the section of lamina propria. The lamina propria, a thin layer of connective tissue, is a part of the mucus. Okay, here is an example of that particular mucus of the mouth. Okay, where These particular IgA molecules synthesized by the plasma cell that is in the laminar propria. Okay. Now let us go through the biological functions of IgA. Acts as antiseptic mucous membrane. Okay, it binds to the pathogenic microorganisms and they prevents the attachment of pathogens to the mucosa of respiratory, gastrointestinal and urinogenital tract. And this IgA provide immunity against tapeworms. This Ig immunoglobulin, which is found in the colostrum or breast milk, protects the baby from intestinal pathogens, helps to protect and nursing newborn. Okay, that is in the intestine, the secretory immunoglobulin attaches to viruses, bacteria, and protozoan parasites such as Entamoeba histolytica, which prevents the adherence to mucosal surfaces and invasions of host tissues. Then what you can say this immunoglobulin as? You can say this immunoglobulin which is providing passive immunity. The immunity is transferred from mother's breast milk to the newborn baby. So you can say this as passive immunity. Okay, natural passive immunity. These immunoglobulin A neutralizes local toxins by promoting phagocytosis. Hope you know the phagocytosis. In previous video, I've explained that. Secretory immunoglobulin found in tears, saliva, and breast milk, right? In these fluids and related body areas, secretory immunoglobulin A forms an immune barrier and plays a major role in protecting surface tensions against infections and microorganisms. Okay, IgA may neutralize various other intracellular pathogens that reside within epithelial cells. Okay, in the intestine, the secretory immunoglobulin attaches to viruses, bacteria, and protozoan parasites such as Entamoeba histolytica. Okay, this secretory immunoglobulin A can also be known as passive, which is going to provide the passive immunity, or we can also say this as naturally acquired passive immunity or passive immunity or maternal immunity. Why maternal immunity? This IgA which is transferred from mother's breast milk to the newborn baby, which provides the immunity, right? The mother provides resistance to a child by direct transformation of antibodies. How they'll transform? That will be transformed through colostrum and the breast milk. Okay, what is this colostrum? Colostrum, which is a thick yellowish fluid produced by breast in first few days after the birth of the baby. Okay, and later this thick milk, thick yellowish fluid will be replaced by mature milk, which suits, that is the colostrum, the child that protects that particular newborn baby, because less of fat and sugar and rich with vitamin A, immunoglobulins, and helps the child against certain infections. Okay, hope you're making note of all this information. If I'm going very quick, what you can do, you can pause the video and you can make a note. Do not waste your time, kindly make a note. And this IgA which also protects from poliovirus, streptococci, diphtheria, tetanus, mumps and rubella. That's why the breastfeeding is very, very important during the early days of the after the delivery of the baby. Okay, here you can see the structure representation of the immunoglobulin. Whenever you take the structure representation, you need to use the terminology immunoglobulin. You will not say the terminology antibody A. Antibody, when are you going to say? During the biological functions, right? Whereas when you are thinking about the structural concept and composition concept, you are using the terminology Ig, that is immunoglobulin. Here in this picture representation, you could see the dimeric form of secretory immunoglobulin A. This is the light chain and this is the heavy chain, first domain, second domain, third domain, and fourth domain. And you need to label this one, right? This is what? This is the variable light chain and constant light chain. This as variable heavy chain, constant heavy chain first domain, constant heavy chain second domain, constant heavy chain third domain, CH1, CH2, CH3. And these are the carbohydrates. And mention here this as, this is the hinge region where you could see the presence of two, here also I have missed here, you need to write here one more disulfide bond. Here two interdisulfide bond, here also two interdisulfide bond, here one interdisulfide, one interdisulfide bonds. Okay, you can label. Apart from what you can label here is that this is the N-terminal region. You can write it here as FAB, that is fragment of antigen binding. Whereas this region from here to here, you can label that as FC, right? Fragment crystallizable. And here we can write this as amino terminal, and the bottom you can write that is the C terminal. Okay. And what is the uncommon here? We can see here this kind of chain. This chain is known as the secretory component, right? And this is the J chain or joining chain. This J chain and the secondary component, which is looking like a S shape, no, both which stabilizes the dimer formation form of secretory immunoglobulin A. So this is about the structural representation of the IgA. You can keep in your mind this particular structural representation also. Okay. Kindly make a note of this one by using pencil. Make a pencil sketch. In the exam, this is very important. Kindly make, this is very easy to write. Okay, this is the overall view of immunoglobulin class of the IgA. The structure is dimer, but that will be in the serum monomeric only. Later that will be dimerized. Okay, percentage serum antibodies, that is 10 to 15 percentage of the all the local pool of the immunoglobulin. Location, that is secretions like tears, saliva, intestine, milk, blood, and lymph. Half-life in serum, that is 6 days. Complement fixation, no. Placental transfer, that is which cross the placenta, no. What are the known functions here? To mention few, localized protection of mucosal surfaces, provides immunity to infant digestive tract. Okay, this is all about the immunoglobulin A. Hope you understood this immunoglobulin A. Right, this is the dimeric form, and this is secretory immunoglobulin, and this IgA, which is going to act like a maternal immunity or passive acquired immunity, right? The immunity will be transferred in the form of the breast milk, where IgA molecule which are present in the milk. Okay. Hope you understood today's class, and you got the information about IgA immunoglobulin characteristics and the structure and functional concept. Okay. I, Dr. Pratibha K, signing off for today. Stay tuned with me. I'll be back with some more videos. And I request you to subscribe this channel if you have not subscribed, and kindly share this video to the needy people. Thank you everyone. Jai Hind, Jai Karnataka.